imagine finding, like, the exact genetic mutation causing a patient's cancer, right?
Yeah.
You engineer this custom billion-dollar drug to destroy it, and then you realize your treatment is just utterly useless.
Oh, yeah. And not because the drug failed, but because, well, the biology is just infinitely more complicated.
Exactly. It's useless because that specific mutation you targeted was actually just a passenger, a genetic hitchhiker that hopped into the back seat long after the crash had already happened.
Which is such a frustrating reality in oncology.
Right. For decades, we've operated on this highly appealing but, ultimately flawed gene-centric model. We look for a mutated gene, we point at it, and we say, that's the villain. But today, welcome to the deep dive, because we are dismantling that illusion for you.
We really are. And we're doing that by exploring this monumental paper published in the journal Cancer Cell. Maria Khalifa and Nicholas McGranahan reviewed a study by Bandlamudi and colleagues that is, frankly, it's staggering in its scope.
Yeah, the scale is wild. They analyzed over 54,300 individual tumors.
54,331 to be exact. And we're talking about a data set spanning 64 different cancer types and 448 distinct subtypes.
Which is mind-blowing.
It is. The sheer scale allows us to see patterns that were completely invisible when we were only looking at, say, a few hundred or even a few thousand cases.
Right. Because uniformly processing over 54,000 tumors strips away the noise. It forces us to look at the disease through a three-dimensional, context-aware framework.
Exactly. The fundamental premise here is that a mutation does not act in a vacuum.
So our mission today for you, the listener, is to completely break down this new framework. No jargon left unexplained. The authors talk about the tissue context, meaning the biological neighborhood entirely changes the rules of the disease. But, before we get into the neighborhood, we have to map the vehicles driving on those roads, right? The mutations themselves.
Yeah, that's the perfect place to start. The paper makes a really hard distinction between two categories of somatic alterations. You have your drivers and your passengers.
Drivers and passengers. Okay, break that down for us.
So a driver mutation provides a direct fitness advantage to the cancer cell. It actively steers the tumor's growth, survival, and evasion of the body's defenses.
It's the one with its hands on the wheel.
Precisely. Now, a passenger mutation is just a byproduct of genomic instability. As cancer cells rapidly divide and, their DNA repair mechanisms break down, they accumulate countless random errors.
Just totally random typos.
Right. These passengers, they don't contribute to the disease progression at all. They just happen to be there.
Which brings us back to that billion-dollar drug design problem I mentioned at the start. Targeting a late-arriving passenger mutation it's like arresting a low-level henchman while the mob boss is still running the entire city.
That's a great way to put it. You haven't actually disrupted the criminal enterprise.
But finding the true drivers isn't always obvious, is it? Because the soil of every tissue is completely different.
No, it's incredibly difficult because, like you said, the tissue context changes everything. A lung cell and a breast cell have entirely different epigenetic landscapes.
Meaning they have different chemical tags on their DNA, right? Things that leave certain genes exposed and others tightly packed away.
Exactly. And because that underlying architecture is different, the specific drivers that are actually capable of taking root depend heavily on that local environment.
So when Bandlamudi's team analyzed the soil across all 54,000 tumors, what did they find? Because the numbers here are huge.
They uncovered 164 novel mutation hotspots.
Wow. 164 new ones.
Yeah. These are highly recurrent, specific genetic sites in known cancer genes that had basically flown under the radar previously. Individually, any one of these hotspots is incredibly rare.
But when you aggregate them...
Right. When you look at the massive data set, 5% of all the tumors analyzed harbored one of these new mutations.
And 5% in the landscape of global oncology, that represents a massive, unexploited therapeutic target for patients.
It's huge. And the data reveals something even more crucial about these 164 hotspots. They're positioned on the evolutionary timeline of the cancer.
Okay, a timeline.
Yeah, they have a strong tendency to be what we call truncal.
Truncal. Okay, so for everyone listening, try visualizing a tumor's evolution as a growing tree. A truncal mutation is one that occurred in the trunk, like the very first founding cancer cell.
Exactly, the most recent common ancestor.
Right. Because it forms the main trunk, that exact mutation is inherited by every single subsequent branch, twig, and leaf of the tumor. It's the ultimate initiating event.
And the mechanics of how these early truncal events operate usually involve what we call tumor suppressor genes.
Okay, let's unpack that. tumor suppressors.
So in healthy biology, tumor suppressors act as the critical brakes on cellular proliferation. They stop cells from dividing out of control. And the paper notes a heavy enrichment for a mechanism called loss of heterozygosity, or LOH, at these specific novel hotspots.
Loss of heterozygosity. Okay, LOH. To wrap our heads around this, think of a tumor suppressor gene like a room with two heavy deadbolts on the door, locking the cancer away.
I like that. The two-hit hypothesis.
Right. You inherit one functional copy or allele from your mother and one from your father. Those are the two locks. If a random mutation breaks one of those locks, you've suffered the first hit.
But the cell doesn't spiral into cancer just yet.
Exactly, because that second lock is still holding the door shut. The brake is still functioning. So what is the LOH part?
Loss of heterozygosity is the catastrophic second hit. It is the secondary event where that remaining healthy functioning copy of the gene is physically lost or deleted entirely.
Oh, wow. So the redundancy is just gone.
Gone. The two-hit requirement is fulfilled, both deadbolts are broken, and the cell initiates unregulated division. And the presence of LOH at these 164 hotspots provides definitive pathogenic evidence that these specific mutations were the foundational events that built the trunk of the tumor.
That is fascinating. Okay, so let's do a quick mini recap for you all listening. We've got drivers steering the cancer, passengers just along for the ride. We found 164 new truncal hotspots acting as the roots of the tree, mostly by smatching both deadbolts on our tumor suppressor genes through LOH.
That's a great summary.
So here's a question for you to chew on before we move on. Think about a time you tried to fix a symptom without understanding the root cause. If you only target a late-stage passenger mutation with a drug, are you actually chopping down the tree, or are you just pruning a little branch?
You're definitely just pruning a branch.
Exactly. Which leads us to a fascinating biological puzzle. If these truncal drivers are the roots of the tree, do certain mutations always act as roots, or can they show up later depending on the soil they are planted in?
Ah, right. Does a timing change? And the data emphatically says yes, it does. The paper classifies cancer genes into two distinct environments to explain this: canonical and non-canonical.
Okay, canonical and non-canonical.
Right. So a canonical context is the gene's home turf. It means the gene is traditionally associated with driving cancer. In that specific tissue type, it's expected to be there.
Got it. Home turf.
And a non-canonical context means the gene is mutated, but it's occurring in a tissue where it isn't traditionally recognized as a primary driver. It's playing an away game.
And the statistic from the study that really emphasizes this is wild. They found that 32% of driver events happen in these non-canonical genes.
Nearly a third.
Yeah. A third of the time, the mutations driving the cancer's progression are operating outside of their traditional biological jurisdictions.
Here's the crazy part. They behave entirely differently depending on whether they are at home or away.
How so?
When a mutation occurs in its canonical context, its home turf, it almost always acts as an early, disease-initiating truncal event. It's the root. But when that exact same genetic alteration occurs in a non-canonical context, the away game, it overwhelmingly emerges as a late subclonal event.
Oh, so it doesn't form the trunk at all?
No. It only appears later in a specific, localized branch of the evolving tumor.
Okay, let's use the case study from the paper to make this concrete, because they used the PIK3CA gene to show this, right?
Yes, PIK3CA. It's a very well-known cancer driver. In breast, uterus, and bladder cancers, which are its canonical home turf environments, PIK3CA mutations are predominantly clonal, meaning early.
They are the founding mob bosses initiating the crime family.
Right. But when that exact same PIK3CA mutation shows up in lung adenocarcinoma or glioblastoma in the brain.
Which are the away games.
Exactly. In those tissues, it is relegated to being a late subclonal event.
Wait, so let me just make sure I have this right. The exact same molecular villain, PIK3CA, acts like the founding mob boss in breast cancer, but it's just a late-arriving opportunist that joins the gang years later in lung cancer.
That is exactly what's happening.
Yeah.
And this temporal shift is a direct reflection of a shifting selection landscape.
Meaning the rules of survival in the tumor are changing over time.
Yes. So in the early stages of a lung tumor, the inherent biological constraints of that specific lung tissue simply might not favor a PIK3CA mutation. An early cell that acquires it wouldn't actually gain a survival advantage, so it just dies out. It doesn't propagate.
But then the tumor grows.
It grows, and it fundamentally alters its own microenvironment. It induces hypoxia, oxygen starvation. It depletes local nutrients. It recruits inflammatory cells.
So the tumor literally terraforms its own neighborhood.
Precisely. And as the neighborhood changes, Those early biological constraints relax. Suddenly, the altered microenvironment creates a specific pressure where a new PIK3CA mutation provides a distinct survival advantage.
Wow. So the non-canonical mutation becomes highly selected for, but only late in the game, in that specific little sub-branch of the tumor.
You nailed it. That's the context-aware framework in action.
All right, time for another quick recap. We've got canonical drivers, which play on their home turf early as the trunk. Then we have non-canonical drivers, playing an away game, showing up late as branches. And the PIK3CA timeline proves that the exact same mutation changes its behavior based on the tissue.
It's all about location and timing.
So listener, if you were designing a drug right now, think about this. Would you prioritize targeting a canonical driver that affects the whole tumor, or a non-canonical driver that might only be driving a tiny resistant subfraction of it?
That's the million-dollar question for pharma companies right now.
It really is. Okay, so we've established that the tissue dictates the timing of the mutation. But what about the patient themselves? Like, does the patient's age dictate the type of mutation that starts the fire?
Yes, absolutely. And Banlamuti's team integrated massive clinical annotations alongside the genomic data to prove this. They specifically tracked the patient's age at diagnosis. And what they found is that the actual mechanics of tumorigenesis changed dramatically across a human lifespan.
Okay, let's look at the skin cancer example from the paper Cutaneous Melanoma. This provides the clearest window into this age disparity, right?
It really does. So the researchers focused on a highly prevalent cancer driver called the BRAF gene. When they looked at older melanoma patients, the driver was almost universally a BRAF point mutation.
A point mutation? Okay, for our visual mnemonic here, a point mutation is like a single typo in an instruction manual, like a spelling cut as cat, just one tiny letter swapped out.
Right. One single base pair out of 3 billion is changed. But when they analyzed younger melanoma patients, the initiating event wasn't a typo at all. It was a BRAF fusion.
A fusion?
Yeah, a gene fusion is a massive chromosomal rearrangement. It happens when two entirely separate genes, often on completely different parts of a chromosome, are physically broken and erroneously stitched back together.
Okay, so if the point mutation is a tiny typo, a fusion is like taking the first half of a Blender instruction manual, ripping it in half, and duct taping it to the second half of a lawnmower manual.
That is a very chaotic but very accurate visual.
It's a massive structural collision of two different genes.
Exactly. You are taking the highly active promoter of one gene and slamming it directly next to the BRAF kinase domain, which basically locks the engine into the on position permanently.
So why? Why does a 25-year-old get the massive Blender lawnmower collision, but a 75-year-old just gets the tiny typo?
It comes down to driver strength versus the immune system. This is profound. It tells us that the threshold required to initiate a tumor is fundamentally distinct across age groups.
Because younger people have better immune systems.
Right. A young, healthy individual possesses highly efficient DNA repair mechanisms and robust immune surveillance. A single point mutation, a typo in a young person's skin cell, is likely caught and eliminated by their immune system immediately.
So the typo isn't strong enough to cause cancer in a young person?
Exactly. To overcome those formidable natural defenses, the cancer requires an incredibly potent, structurally massive event like a fusion to tip the cell into malignancy. It needs a stronger driver.
Oh, man. That makes so much sense. Meanwhile, the older patient's cellular landscape has been bombarded by decades of environmental damage, right? Like UV radiation from the sun.
Yes. Their immune surveillance is naturally waning. The barrier to entry for the cancer is significantly lower.
So the local environment is already so degraded that a simple single-letter typo in the BRAF gene is enough to ignite the disease.
Exactly. The strength of the driver required is entirely dependent on the age and the resilience of the soil it's planted in.
That is so elegant. All right, recap number 3. We looked at clinical annotations by age. We saw that early onset melanoma relies on massive BRAF structural fusions to overpower a strong immune system, while late onset melanoma just needs a simple point mutation because the body's defenses are lowered.
Perfect summary.
So think about how our bodies age for a second. Consider why a younger immune system requires that massive structural collision to trigger cancer, while an older body might succumb to a simple genetic typo. It really reframes how we view aging and disease.
And it perfectly sets up the ultimate frontier of this entire framework. Because mapping the structural drivers, the timing, and the age-related mechanics, that's really only half the battle.
Right, because eventually we have to figure out how to actually fight the cancer.
Exactly. The true complexity arises when we attempt to intervene therapeutically, and that requires us to understand the immune contest, specifically precision immunotherapy and immune evasion.
Which is easily the most fascinating and honestly terrifying part of this deep dive. Let's talk about T-cell receptor therapies, or TCR therapies.
So your T cells are the primary enforcers of your immune system. They are the attack dogs. In TCR therapy, scientists engineer these cells to hunt down and destroy cancer.
But a T-cell can't just attack blindly, right? It needs a specific target.
Right. It's looking for what we call a neopeptide. When a cancer cell harbors a mutation, like in the KRAS or TP53 genes we talked about, it produces abnormal mutated proteins.
The neopeptides.
Yes, and these neopeptides are completely unique to the cancer cell.
Okay, visual analogy time. Think of the neopeptide as a highly specific wanted poster. It features the exact molecular face of the criminal cancer cell. But the T-cell cop can't just spot this wanted poster floating around inside the cell or lying on the ground.
No, biology doesn't work that way. Biology requires a presentation mechanism.
Right, the wanted poster must be physically pinned to a very specific kind of molecular bulletin board on the exterior surface of the cancer cell. And that bulletin board is called the human leukocyte antigen, or HLA complex.
The HLA complex. If the neopeptide isn't presented on the HLA complex, the immune system remains completely blind to it. It just doesn't see it.
And this is where the paper reveals the absolutely cruel math of immune restriction. The researchers found that roughly 14% of patients harbor the specific KRAS or TP53 mutations capable of producing these wanted posters.
Which, from a therapeutic standpoint, looks like a massive population eligible for TCR therapy. You think, great, 14% of patients, we could treat them.
But the reality is a fraction of that. Because having the wanted poster isn't enough, you have to have the exact right bulletin board to display it. And the shape of your HLA bulletin board is dictated entirely by your genetic ancestry.
It's genetic roulette. The study found that of the 14% of patients who actually have the mutation, only 22% of them possess the specific HLA alleles required to present that specific neopeptide.
Oh, wait, only 22% of the 14%?
Yes. This is called HLA restriction. If you were in the 78% of patients whose genetic ancestry provided a differently shaped HLA receptor, the neopeptide won't fit. The poster cannot be pinned to the board.
So the cutting-edge immunotherapy will fail?
It will fail. And not because the drug didn't work, but because your specific immune context, your ancestry, cannot physically present the target to the engineered T cells.
That is brutal. And as if that genetic lottery wasn't challenging enough, the tumors themselves are fighting back, aren't they?
Oh, they are. The Bambamutti analysis revealed that 25.4% of tumors exhibit a copy number loss of HLA.
I'm sorry, what? 25.4%?
Yes. Over a quarter of these cancers are literally operating without bulletin boards.
So the tumor literally realizes it's being hunted, and it responds by tearing down the bulletin boards so the wanted posters can't be displayed.
It is a stunning example of immune escape. But, and we have to be careful about how we frame this, the tumor isn't conscious.
Right.
It isn't realizing it's being hunted and making a decision to tear down the boards.
Right. It's pure Darwinian evolution by subtraction.
Exactly. The selective pressure is immense. The immune system is constantly patrolling. Any cancer cell that successfully displays a neopeptide on its HLA complex is immediately identified and destroyed by your T cells.
Oh, I see. Therefore, the only cancer cells that survive to divide and replicate are the rare, random mutants that just happen to experience a genetic deletion of their HLA complex.
Exactly. The immune system inadvertently selects for the invisible cells. By wiping out the visible ones, it leaves behind a highly aggressive, immune-evasive subclone that has effectively deleted its own presentation machinery.
It's evolving into a shape-shifting enemy. And the paper points out they don't always even delete it genetically, right? Sometimes they use non-genetic ways to hide.
Yeah, epigenetic silencing. They just downregulate the HLA expression, effectively dimming the lights on the bulletin board without genetically deleting it.
Unbelievable. Okay, fourth and final mini recap. We've learned about neopeptides acting as wanted posters, TCRs as the cops, and the HLA bulletin boards. We learned that ancestry dictates if your board is the right shape, and that over a quarter of tumors use immune escape to literally tear down their own boards to hide.
It's a daunting landscape.
It really is. So, listener, if a quarter of tumors are deleting their own HLA to hide from our therapies, how must our treatment strategies adapt to corner an enemy that literally shapeshifts?
Yeah, is the big challenge for the next decade of oncology. And understanding that evolutionary pressure really highlights the limitations of how we currently profile these tumors. which the authors of the review, Kaliva and McGranahan, critically address.
Let's talk about those limitations, because as massive as the Bandler-Moody study is, it's not perfect.
No study is. The greatest strength of this study is its sequencing depth and the use of matched normal DNA samples for all 54,000 patients. That allows for incredibly precise variant calling. We know exactly what mutated.
But the weakness is the lens they used, right? They relied on what's called targeted panel sequencing.
Right. They were looking at a highly curated panel of up to 505 known cancer-associated genes.
So by only looking at a panel of 505 usual suspects, we are entirely missing the completely undiscovered or de novo cancer genes that a whole genome sequencing approach, WGS, would catch.
Exactly. It's the streetlight effect. We're only looking for our keys under the street lamp because that's where the light is. The targeted panel gives us a high-resolution map of the houses, but it doesn't show us the infrastructure of the entire city.
We miss the structural variants in the non-coding regions.
Yeah. The authors stress that the future of precision oncology cannot rely on genomics alone. To truly understand the context that Bandler-Moody's team proved is so vital, we have to integrate multi-omic data.
Multi-omic, meaning multiple layers of biology.
Yes, we need transcriptomics to see what RNA is actually being expressed. We need epigenomics to map the chemical tags turning genes on and off. We have to map the entire neighborhood, not just the genes.
Bringing it all together, the ultimate takeaway from this massive data set is a total paradigm shift. Yes, the paper notes there are rare instances where a therapy works completely independently of tissue context, like using immune checkpoint inhibitors in tumors with widespread mismatch repair deficiency.
But those are the exceptions.
Exactly. The rule, overwhelmingly proven by these 54,000 tumors, is that the consequence of a genetic mutation is inextricably linked to both the specific gene and the complex biological neighborhood it arises in.
We can no longer look at a pathology report, see a PIK3CA mutation, and reflexively prescribe a PIK3CA inhibitor.
Right. We have to ask the deeper questions. Is this mutation canonical to this tissue? Did it happen early in the trunk or late in a branch? How old is the patient's soil? And does their specific genetic ancestry even allow their immune system to see the mutation?
The definition of precision oncology has to evolve. It's moving from a rigid, one-dimensional focus on just the gene into a fluid, three-dimensional understanding of spatial and temporal biology.
Which leaves us with a profound, almost philosophical shift in how we approach treatment. And this is my final provocative thought for you to mull over today. If a tumor's behavior, its evolutionary path, and its vulnerability are dictated entirely by its local environment, by its epigenetic landscape, and local immune constraints, are we reaching a tipping point in medicine?
A tipping point where we don't just attack the cell.
Exactly. Perhaps the future of treating cancer isn't solely about deploying toxic agents to hunt down and destroy the diseased cell. If we can develop epigenetic therapies that rehabilitate the neighborhood, if we can therapeutically alter the soil of the tissue itself, can we effectively strip a canonical cancer driver of its license to operate?
It's a complete inversion of the war on cancer. Defeating the tumor not by killing it directly, but by changing the environment that allows it to survive.
It's an incredible thought. We appreciate you joining us for this intensely detailed deep dive into the molecular architecture of cancer. The source material was heavy, but understanding these mechanics is how we push the boundaries of what's possible.
Thanks for exploring it with us.
Keep questioning the context of the information around you. Until next time, keep diving deep.